Hello children, how are you? Fine? Hope you are enjoying my video class. So today I shall be taking the second chapter of your 12th biology NCERT textbook, that is the topic sexual reproduction in flowering plants. Now when we talk about sexual reproduction of flowering plants, the first thing comes to our mind: what is the reproductive part of a plant? By name itself, the flowering plant, you can easily guess, flower is the reproductive part of a plant. Flower. So what is flower? We can define very simple way. Flower is a modified shoot, helps for the process of reproduction. So flower is a modified shoot which helps for the process of reproduction. Now let us go in detail about the structure of the flower. If I draw a very simplified diagram, this is the innermost or central part of flower. This is the second very important part of a flower. And one side I am drawing only. The third one, and the fourth one. So one side I have drawn. This is this entire part, what you call as female reproductive part, that is commonly in general we call as gynosium. This is the male part called androcium. This is and this other one, the petal, and this one, the sepal. So this sepal and petal, they are the accessory reproductive part. They are not directly involved for the process of reproduction, but they help to protect the innermost part, that is androcium and gynosium, and also the petal especially attract the various insects and other animals because of their beautiful color and nectar. So the fragrance and helps for the process of pollination. So let me wrap these two parts, the leaving behind the androcium and gynosium. So the androcium or male part, otherwise called stamen, and this gynosium called carpel or pistil. In androcium may composed of so many stamen if the number of stamen is large. In that case, sometime the entire stamen can form a single bunch or can form two such bunch or three or more than that. If it is forming a single bunch, then it is called monoadelphous. The terminology we have read in class 11 standard: monoadelphous, diadelphous, polyadelphous, when many such bunches form. In case of gynosium, the unit pistil or carpel, the gynosium can be consist of one carpel, monocarpellary, or the ovary part, or the polycarpellary, whatever it is. If single, more than one carpel, then it can be jointed with each other, called syncarpus or free apocarpus. This terminology you have already read, but just a recapitulation of the previous lesson, previous session. Now let us go in detail about the androcium and the gynosium, or the pistil and the stamen. So we are now entering to the pre-fertilization structural event. Why it is the name given pre-fertilization structural event? It is because before the process of fertilization takes place, the various events required to facilitate or help the process of fertilization, that is what we call as pre-fertilational events. And the structure associated with the fertilization, that is pre-fertilization, and then the fertilization will come, that is the androcium and gynosium. So let me take androcium, that is stamen. So shall I rub the board and just go for the next? Okay. So if I take up a single stamen and draw a very simplified diagram, a stamen which is having a stalk-like part like this and having anterior end of this stalk a bag-like structure. So this stalk-like part, what we call as filament, and this is called anther. So the anther is a bilobed. You can see here two lobes. It's a bilobed structure. So anther is a bilobed structure. Now, this bilobed structure of anther, having a longitudinally from one end to other end, runs a groove. As a result of this groove, each such lobe of anther divided into two parts, one this and another this. This is what we call as now, so same as the case for here also, this lobe. So as a result of that, this groove, what we call theca, because of that presence of theca, now the anther, which is actually a bilobed structure, now divided into four compartments, four lobes. So if I draw a transverse section, how it will look? It will look like this. It will look like it will almost look like this. Almost look like this. having this tetragonal structure. At each such tetragon, each such almost circular structure of the tetragon, at each corner of each such gone, there lies one microsporangium. What is it called? Microsporangium or microsporangia. This microsporangia runs throughout the length of the anther lobe, and when gets matured, it is converted into a pollen sac. What is it? Pollen sac. Pollen sac. So, if you cut a transverse section, it will look almost like this. The microsporangium, which further develops round through the length of the pollen, length of the anther, and when matures, they gets converted into pollen sac. Now, if I draw a cross section from this part, if I draw a cross section, how it will look? How the microsporangia will look? It will look almost like this. It is composed of a 4-layer of wall from outside to inside. If I draw, how it will look like? Try to concentrate here. Very simplified diagram I am drawing. The outermost layer, so only one I am drawing here. Remaining all the lobes are having same structure. So let me draw another lobe. So this wall or this layer is called as epidermis. Epidermis. Next, second layer this layer is called endothecium. This layer is called endothecium. So outermost layer epidermis, second layer endothecium. Endothecium. Next layer of cells this is called middle layer. And the innermost layer, which is densely packed with cytoplasm and having nucleus, most of the cases more than one nucleus. So this densely packed with cytoplasm and having nucleus, This is the innermost layer, what we call tapetum. So this layer, please concentrate here, this layer what we call as innermost layer called tapetum. Tapetum. So these three layers, epidermis, endothelium and middle layer, they are just acting as a protective layer. Epidermis, so let me be very specific. So the outer three layer, what are those? Epidermis, endothelium, middle layer, they are just meant for giving protection. These three layers are meant for protection. But the innermost layer, which is dense with cytoplasm, having dense cytoplasm, and a centrally placed nucleus. Most of the cases more than one nucleus. Although I have drawn here one nucleus. This is called tapetum. This tapetum is very, very important. Tapetum. Because it is the nourishing tissue. Give nourishment. It is the nourishing tissue. To whom it will nourish? To the innermost cells. Innermost cell mass. So if I draw the innermost cell mass in case of a young when it is very young, in case of a young microsporangia, the innermost cells almost looking circular, innermost, which is made up of a compact layer of cell, These cells together forming the tissue called sporogenous tissue. What is it called? Sporogenous tissue. Sporogenous tissue. In case of immature microsporangium. When the microsporangium is immature, it is called as sporogenous tissue. The innermost cells forming the tissue surrounded by the tapetum, these innermost cells are called sporogenous tissue. These sporogenous tissues are actually, each and every cells of this sporogenous tissue are actually a functional microspore mother cell. So they can be also written as microspore mother cell. Is it very clear? Now, when the sporogenous tissue, cell of the sporogenous tissue are getting mature, they require a lot of nutrient, that is nourishment is to be given. Who will give the nourishment? It is the tapetum. Who will give the nourishment. So each and every sporogenous cells of the sporogenous tissue are actually a functional microspore mother cell, MCC. Is it clear to you? Now, once we know the structure, now we shall come to the next point, that is microsporogenesis. So shall I write this part? Microsporogenesis. So what I shall discuss? Microsporogenesis. What is microsporogenesis? How can you define microsporogenesis? It is the process of formation of microspore from microspore mother cell. So the process of formation of microspore from microspore mother cell. What is the microspore mother cell? Here. So these cells which are diploid having 2n number of nucleus will undergo division. What type of division? Because microspore is haploid, so it has to undergo a division. That division, cell division, is meiotic division or meiosis. So the process of formation of microspore from microspore mother cell is called microsporogenesis. Microsporogenesis. Now, let us discuss the process of microsporogenesis. So, shall I wrap this part? You have all very clear, you got very clear idea. So, let me wrap this part. What is microsporogenesis? Now, the cells of the microspore mother cell or the sporogenous tissue during microsporogenesis the cells of microspore mother cell will divide meiotically, meiosis. So during meiosis, two divisions take place, twice the cell divide. So in first division, it forms two cells, then second division, so ultimately gave rise to a cluster of 4 cells. This is now called microspore tetrad. Why it is called microspore tetrad? Because of tetra means 4. So the 4 microspore, each one is a microspore, each one is a microspore. So by means of microsporogenesis from one microspore mother cell, by means of meiotic division, 4 microspore are formed and they arrange in cluster. These clusters of 4 microspore we call microspore tetrad. Now, when they get mature, then each microspore tetrad separate from each other, and now they are called pollen grain. So in microspore tetrad, how the microspore tetrad form? By means of meiotic division. From where it will form? It will form from the sporogenous tissue of the microspore mother cell. Where does it present? It presents at the central part of the Microsporangia, which is guarded by 4 layer of wall, that is tapetum, middle layer, endothecium and epidermis. Out of that, it is the tapetum which nourishes the microspore mother cell. From where, by means of microsporogenesis, microspore tetrad form, and this tetrad, when gets mature, dissociate from each other. Thereby, the pollen grains are developed. Is it very clear to you? Now, if you go to my early diagram, within the pollen sac, Now this pollen grain, if you touch a mature pollen sac, your fingers will be filled with almost yellowish color grain, small microscopic grain-like structure, dust, powdery dust-like structure. That is nothing but the pollen grain. If you study a bit detail about the structure of pollen grain, let me rub this part. Now the pollen grain, if you collect the pollen grain from different flower samples of different species, you will get a variety of structure. Some are big, some are small, some are having smooth outer covering, some are having wavy outer covering. But one thing is common, in all the cases, the pollen grain is having two layers of covering, the outermost layer called XII and the innermost layer intine. The XII is made up of a very hard substance. Sometime previous discussion also I have told you that is called sporopollenin. This is made up of sporopollenin, which is very highly resistant against the action of enzyme, acid or base, which helps to protect the pollen grain. Now when the pollen grain dissociate from each other after maturation, it landed over to the stigma of the female part. Generally by that time the nucleus, pollen nucleus divide, cytoplasm also divide to form a two-cell state. The upper one is a bigger cell, what you call as vegetative cell. And the lower one, a small one, as if it is suspended within the vegetative cell, cytoplasm, that is called generative cell. In this condition, that is two-cell structure, at this two-cell structure, generally pollen grain landed over the stigma of female flower. That is the case of around 60% flowering plant. Remaining 40%, they generally remaining 40%. So what I can tell you, in case of 60% cases, when the pollen grain landed over the stigma of female flower, female part, they are at two-cell stage. The bigger one, vegetative cell, and smaller one is a generative cell. But in remaining 40% cases of pollen grain, before landing over the stigma, the generative cell divides into two cells. So now it is a three-cell structure. So in 40% cases of pollen grain, it lands when it is a three-cell structure, be very, very specific. But 60% cases, when it land when it is a two-cell structure. Is it very clear? So most of the cases, the pollen grain landed over stigma at two-cell structure. After landing, the post-fertilization, the next, after pollination, the next event will take place, that is through the germ pore, that is the small opening where there is no sporopollenin, through which the entire will come out in the form of a small tube. We shall discuss in next class. For the time being, one more information I would like to share you. This pollen grain is very allergic in nature. It can cause even severe to mild form of allergy, bronchitis or asthma in many people, not all. Especially some pollen grain, like if you might have heard about carrot grass, that is Parthenium. Why it is called a poisonous plant? Because the pollen grain is very allergic, causing asthma. So inhalation of this pollen grain is very harmful. But on the other hand, the pollen grain is very useful also. Pollen grain is highly nutritive. That is why it is also nowadays used as a pollen pollen tablet, and or some cases it is used as a pollen syrup for boosting the body metabolic activity. Especially athletes are using this pollen tablet. This pollen grain, as far their viability is concerned, after the pollen grain is, the pollen tube is burst and the pollen grain is released, how long it will survive? Depends upon again species of plant. Say for example paddy, it approximately remains survive for say half an hour. Or in some cases like family like Solanaceae, like your Rosaceae, like your Malvaceae, these cases of family, the flowers, when the pollen is released, they can survive even up to 1 month. But one thing is sure, this XII is very helping for the pollen grain to its existence. But the pollen grain, just like sperm bank you have heard sometime, can also be preserved in pollen bank. Why? For future use, as and when we require. In that case, by a technique called cryopreservation. What is it? By preserving it in liquid nitrogen, having a temperature of minus 196 degrees Celsius. So the pollen grain can be preserved in liquid nitrogen for longer period of time. So I am sure and I believe that this thing is very clear to you, got a very clear idea up to this pre-fertilizational event and structures we have discussed today up to microsporogenesis and the structure, internal structure of the microsporangium. Till then, meet again in next class, and till then, bye.